Thanks, Karen, for your overview of CMC considerations for cell and gene therapy products. This talk will focus on the non-clinical assessments of CGT products to support an IND. What I hope you will learn from this talk are the regulations that mandate non-clinical testing, what are the non-clinical considerations for early phase trials, links to relevant guidance documents, and a discussion on when to have interactions with the FDA for your non-clinical program. Title 21, Part 312, Section 23A8 of the Code of Federal Regulations mandates non-clinical testing. This regulation outlines the details of pharmacology and toxicology assessments of a new drug required as part of an investigational new drug or IND application. It is important to note that the kind, duration, and scope of such assessments varies with the nature of each proposed clinical investigation, implying that the non-clinical evaluation for each product is determined on a case-by-case basis. In the life cycle of a drug development, non-clinical studies typically come in at early stages before initiating the phase 1 clinical trial. What sort of data can support the initiation of a phase 1 clinical trial? This can be data derived from multiple sources, such as GLP or non-GLP, well-controlled non-clinical studies, published data in peer-reviewed journals, cross-reference to similar products in previous submissions, as well as data from clinical trials, can all be used to support an IND. The primary expectation from a non-clinical program is to demonstrate the safety of the CGT product through well-designed toxicology studies. The goal of toxicology studies is to determine the no observed adverse effect level, or the NOAEL as it is called, for the product, and evaluate any unintended activity of any component of the product, such as the cells, vector, transgene, or packaging elements. Such unintended effects could include presence and activity in any off-target tissues, any local or systemic effects seen either acutely or in the long term, potential risk of immunogenicity or tumorigenicity from the product, or any unintended effects of the drug delivery systems used. While we are on toxicology studies, I do want to emphasize that as per 21 CFR Part 312, each toxicology study should be performed per Good Lab Practices or GLP. If this cannot be done, an explanation should be provided, and all non-GLP studies must have an oversight by an independent quality assurance person or team. In addition to demonstration of product safety, the non-clinical program is also expected to demonstrate the activity of the CGT product in vitro and, when feasible, in vivo in a relevant animal species or any other alternate test systems. Data from such proof of concept studies, along with findings from toxicology studies, should together inform about the optimal dose levels and dosing regimen to be used in the proposed clinical trial. Cell and gene therapies have several potential safety concerns, which can either be associated with the product itself, as listed on this slide, or the procedure that is used to deliver the product. Ideally, the product administered in non-clinical studies should be identical to or as similar as possible to the intended clinical product. For any animal-derived analogous products, it is important to characterize the level of analogy between the human and the animal product and clearly justify the human equivalent dose or HED calculations. It is critical to use an appropriate animal model for non-clinical studies. The animal model used should be permissive to the product and/or recapitulate elements of the target indication. Therefore, there is no default animal model, and scientific justification should be provided for each species or model used. This slide gives some pointers to help select the most appropriate animal model to assess the safety and activity of a specific CGT product. We will now spend a few minutes on non-clinical study design considerations, which is the key to get accurate results from non-clinical studies. A robust study design is non-biased, mimics the planned clinical scenario as closely as possible, includes appropriate controls and multiple dose levels of the investigational product, and includes adequate animal numbers in each study group, all while incorporating the 3 R's for each study. A robust study design is also of a sufficient duration, incorporates multiple time points for in-life assessments, and includes multiple assessments, both in-life and at necropsy. Such a robust study design is key in accurate evaluation of the long-term safety and activity of a CGT product. Some of the key assessments of safety and activity of a CGT product which should be tested at multiple time points include functional outcomes, kinetics of biodistribution, tumorigenicity, transgene activity, and immunogenicity. Toxicology assessments include mortality, any clinical changes including body weight, and a comprehensive evaluation of adverse findings in each animal in each study group. As discussed by Heather and Karen, we encourage sponsors to communicate with the FDA from the earliest stages of product development via the INTERACT and Pre-IND meeting pathways. From a non-clinical standpoint, an INTERACT meeting would be at a stage when the sponsor has generated preliminary safety and proof of concept data, but has yet to initiate definitive safety studies. Guidance from CBER at this stage should help the sponsor design robust safety and toxicology studies for their specific product. The goal of a pre-IND meeting, on the other hand, is to achieve a successful IND submission. From a non-clinical standpoint, this is a stage when all proof of concept and preliminary safety studies have been completed, and the sponsor is ready to conduct definitive safety studies. A pre-IND package should discuss all the elements of a non-clinical study design that was discussed previously, such as animal models used, dose, regimen, endpoints, sacrifice intervals, the rationale for using each of those elements, and the resulting data interpretation. To summarize, the non-clinical program for any CGT product is determined on a case-by-case basis. Non-clinical data submitted in the IND should support the safety and biological activity of the CGT product in the proposed clinical indication. There are multiple opportunities to obtain early feedback from the FDA on non-clinical development plans prior to IND submission. It is important to note that novel therapies mean novel testing paradigms, and hence pre-submission interactions with the FDA is highly encouraged. So come to us early. These are some relevant guidances to help design non-clinical studies for CGT products. And here is my contact information if you have any questions about this talk. Thank you all for your attention.